points. And maybe some of y'all have even actually already had that in high school. So definitely not a fancy college level piece of material at all. It'll be super easy, super swift. Okay. In the lab section, I have all the models, a PowerPoint that I've made of all the models from heart to development. Okay, so when you go to the lab section, you'll see a PowerPoint that has a picture of everything. Because I had a feeling that they were fixing to kick us out, so I just went to the back and I just took a picture of everything and downloaded it from my phone, and or I guess uploading, whatever that's called, and made a PowerPoint of it. So we have boards like this on the left that have all the models that you need to know. And then it's the next slides, they'll be all broken down. So if you ever want to like get a feel of a start to close enough to finish, come back and look at this board so you can see what the steps are. All right. I showed you the top left one already with the sperm and the, I think, with the sperm coming in and meeting the egg, and then you have a zygote. So when the sperm does meet the egg, meet the ovum, which is a secondary oocyte, by the way, then the ovum will finish the second meiosis, and it'll throw off another polar body. So just going back through meiosis with the female, so we'll have these primary oocytes that get recruited once a month. Only one of those guys is going to make it, and they all have a primary. They have a primary follicle, and they all have a primary oocyte, and they all start to mature and develop. Only one of them is going to get chosen to fully develop into a Graafian follicle, and it will be a secondary oocyte. It is not going to finish the second meiosis, though. With ovulation, it will become, it'll get stuck in the second meiosis, and only if that egg gets fertilized will it finish, and it'll throw off the third or the second polar body if the egg is fertilized, if the ovum is fertilized. So when you have an egg that is fertilized, then you have the 23 chromosomes from the male and the 23 chromosomes from the female. They come together, and that's 46, and you have a single-celled zygote. Very quickly, that single-celled zygote is going to start to go through mitosis. And as it goes through mitosis, eventually what's going to happen is we're not just replicating a zygote. We're going to start to go through something called differentiation, which is going to start to make the cells not so much of everything. It's going to separate them into layers, which will determine which route they can go. So they differentiate into 3 different layers. And if you remember from the connective tissue section from AMP1, for example, all of them come from the mesoderm. So I'm going to go through that probably more than you ever wanted to know. But let's go ahead and start with this picture here on the right. So we're going to go through mitosis, and 1 cell is going to become 2. 2 cells are going to become 4. 4 cells are going to become 8. 8 cells are going to become 16. And 16 cells become 32. All of that division is called cleavage. So they're very rapidly going to divide. Once we get into the 32 cell stage, which is the bottom right picture here, it looks kind of like a blackberry or a raspberry. It's called a morula, and that is actually Greek for berry. So that's how he got his name. Once it gets into that much division, he can't, there's getting too much pressure. It's getting too crowded in there. So what's going to happen is that morula, a couple of things. The morula at this point is going to reach the uterus, and this is about day 7 to 10. It's going to reach the uterus, and it's going to seed into the endometrium. The way that it seeds is the outer rim of cells that you see here when you're looking at the right-hand picture on the top left of the actual model. The cells that are going all the way around, and you see the little hollow thing in the middle, the cells that are going all the way around are going to be called trophoblasts. And the little clustered group of cells there is called a blastocyst. The pocket that's going to be all full of fluid, so this is going from morula, which is the bottom right, to the top left, where it's a small circle, but you can clearly see that there's a pocket. The cells on the outside are called trophoblast. The little group of cells that are kind of clustered to the right-hand side, those are called blastocyst. And then the little pocket of fluid is called a blastocele. C-O-E-L-E, blastocele. Okay, so what's going to happen is that trophoblast is going to make these little bitty fingers. And the little bitty fingers are going to seed into the endometrium, and it's going to set up house. Actually, and this is the endometrium, and what it's going to do is it's going to proliferate and get fluffier and fluffier and fluffier. It's actually going to end up hiding this little new baby that's forming right here. So if you were to take a scope and look into the uterus and actually look at this point for that now embedded baby, you're not going to be able to see it. But we know that it does a lot of differentiation at this point. So those little trophoblasts are going to make some little indentions and burrow itself into the endometrium, and that's called villi. This is a little bit too far advanced, but I mean from the next part that I'm going to talk about. But the villi that are going into the endometrium and the trophoblast that's remaining, it's eventually going to differentiate into something called the chorion, and then that is going to become something called the placenta. You've heard of that. And that guy's job is to make sure that right now embryo, but at 8 weeks fetus is getting all the nourishment it needs. It's going to create the blood link between mom and baby. So once we get to the big circle, I can't go backwards or else it'll erase my recordings, but the big circle with the bigger pocket, so on the model on the right, it was on the bottom left. Where all those cells are, it's going to change names. Instead of being called a blastocyst, it's going to be called the inner cell mass. And that inner cell mass is going to start to go through some differentiation and eventually will become the baby. This change, as it goes into this these layers that you see right here with the yellow and the purple with the line in the middle, that's called gastrulation. This is going to happen at about day 10 to 14. So remember, it just took about a week. Everything got fertilized in the oviduct / uterine tube / fallopian tube. So the sperm met the egg up in that tube, and then by day 7, it has implanted into the endometrium. By day 10 or 14, it's going to go through these changes that inner cell mass actually develops layers. So it's going to end up developing into 3 separate layers. Now when it's got these 3 separate layers, now instead of being called an inner cell mass, it's going to be called an embryonic disc. And that embryonic disc is very quickly going to be obvious 3 layers of the ectoderm, the mesoderm, and the endoderm. And I hope if I go to the next slide, it'll be there. I think it is. Yes, it is. Okay. So that plaque that was on the left-hand side of the first slide, this is the first model of the embryonic disc. So what you're seeing here is 3 different layers. They are very quickly going to continue to develop into things that we know of. So endoderm is going to become the digestive system, lots of organs. The ectoderm is going to become skin derivatives like sweat glands, and the neural tube, which is going to end up being the spinal cord and the brain. Isn't that crazy? Your brain and your spinal cord, which is so deep, and then your skin, which is so external. But the ectoderm becomes those 2 things. The mesoderm is in the middle, MM, and the mesoderm is what you already know to do the connective tissue, but it does some other things too. It does muscle, and it does the skeletal system. So all this differentiation, within just a few more days, we're going to end up having this thing on the right. So this picture on the right is actually showing those little blocks right there are called somites. And let's see what I'm going to say about that. The somites, we're at about like week 3. So past 14 days, we're about week 21. Not 21, 12. Do I have dyslexia? What the heck? That little line that's going straight down the middle of it right there, that's called the neural groove. And that's actually going to be the area that the spinal cord passes through. The somites, which again came from the mesoderm, that's going to become vertebrae, skull, even muscle. Isn't that weird? That's weird. Those reddish kind of things on either side, that's the developing heart. And actually, that guy is functioning. All of this is going to fold up and meet the sides. I mean, the right and left side are going to end up folding up. If you can appreciate, if we've got something called a neural groove, and then on either side of it is the neural fold, the elevated parts, that's all going to come together, meet each other superiorly, and form a tube, a passageway for the spinal cord. Those 2 little red bumps very quickly are going to meet, and that's going to end up becoming the heart. The heart is functioning at the end of week 3. It is a functioning organ. It's one of the first organ to function, which makes sense, right? I mean, we need something to be pushing blood all around this new baby. All right, so this is going to be week 4. Big difference, right, between that last picture and this picture. So the 2 ends have come together. You can actually still see those somites on the right-hand side there, but both little blocks, those little blocks called somites, have curled up and come together. At this stage, week 4, we've got a lot of organogenesis coming on. So we're making all kinds of organs. The little purple part right there is going to become the liver. The pink part is going to be the heart. On these different little bumps up by the, what you can clearly see is going to be the head. The first little bump is going to be the maxilla. The second little bump is going to be the mandible. And actually that third bump is the hyoid bone. Isn't that crazy? It's amazing and crazy. So now we're going to start to get, as these guys come together, we're going to start to get at week 4 that cartilaginous skeleton that you learned about in AMP1. Remember, most long bones have a cartilage model first. Muscles are going to start to appear, and you have this little tail-looking thing. So this is like week 3 to 4 is when all of this stuff happens. You're getting all your organs, and you can actually start to see that this is a little person. Now, caveat: if you looked at a dolphin at about this stage of development, it too would look just like this. Oops, darn it, on that last one I forgot to tell you about the little arm and the leg buds, but I think it's labeled in your book. Is it labeled in your book? Yes, it's labeled in your book, although I might be missing a model. On my book, on page 274, yeah, I am missing a model. No, it's this picture right here in the middle of this amniotic sac. Ta-da! That's it. Okay, so right now this is about 5 weeks. You can see a little where they've outlined in black. That's called an optic disc. You can see little buds coming out. This actually might be a little bit further along, maybe 6 weeks. But you can see a little arm and little leg clearly developing. You can see the umbilical cord. So what was trophoblasts, and that became chorion, and all around it, you see that pink? That is placenta forming right there. And then that yellow thing, I think it says number 38, that's a yolk sac. And a yolk is totally different in people than it is like for a chicken. So in chickens, yolk is all about nourishing the chick. This guy, what he's going to do is he is making that little baby's blood, which eventually the spleen will take over and then the bone marrow will take over, right? But early on, in the first month or two, we have this little yolk sac. He's making that baby's blood. Another thing that guy is doing is he is actually, he has the germ cells that's going to decide if this baby is a male or female. I guess actually genetics decide that, but the germ cells are actually in that yolk sac. Remember I said in the last little chapter how we had homologous structures. So if you did an ultrasound really early on right here, there is no differentiating this baby between male and female. The purple thing around that baby is the amniotic sac. I think I mentioned that. And inside there is amniotic fluid, and that is protecting the baby, keeping the baby moist, compartmentalized, giving it kind of a cushion so it can bounce around. So that's all cool. And then the picture on the right, you can really just tell that that is that's a full-formed baby. I mean, we've got maturing to do, but clearly that's a little peanut. At this point, that chorion has developed into a placenta, which is taking up the majority of the rim of that uterus right there. You can see the white cervical plug, so that's keeping any sort of bacteria out, keeping it away from the baby. Okay, the one on the left, that baby is about 1 month. The one on the right, that baby is about 2 months. Now notice on the one on the left, you see a number 2, and that's the placenta, and you can see it wrapping all the way around. When we move on to 2 months, then you can see the placenta is kind of squishing over to one side. So that's about 2 months. At the end of the 2nd month, all the organs are formed. They might not be all working, but heart's working great, liver's working. We're going to start to get real bone forming at this point. And at 2 months, at 8 weeks, we're not calling it an embryo anymore. We're calling it a fetus. It looks like a little human. And at 2 months, a little bit more than 2 months. Now all the organs are starting to be perfectly working. This is 3 months. Look at that placenta. It's kind of squishing over to the side there. A couple of other things I want to talk about. We have lots of changes starting here. So at 3 months, moving on to 5 months, we're going to have lots and lots of growth. Hair is going to start to show up on the baby's head and body. And there's actually, the baby is going to be covered with a fine hair called lanugo. And if you look at my lecture PowerPoints, you'll see a baby that was born prematurely, and you can really appreciate how the baby just looks like fuzzy. And they call it, in your lab book, they call it downy hair. But, well, they call it lanugo, but they say it's like fine downy hair. But it just looks fuzzy. My first baby was born at 31 weeks, and she definitely was fuzzy. Also, babies are born with this coat of something called vernus caseosa, which is going to start to become obvious after the 5th month. And it's like secretions from the sebaceous glands of the baby. So sebum, it's kind of greasy and oily, and then dead skin cells, because the baby's changing so much. So baby's growing, so is its skin, and it's sloughing dead skin. Remember, skin changes once a month, and I honestly don't know how quickly it changes in a baby or in a fetus, but it's probably faster than that because they're having such rapid changes. The first time that baby starts to move is usually going to be right after 5 months, so about 20 weeks, and the mommy's going to feel it. It's going to feel like a little flutter or maybe some gas moving. As a matter of fact, you might think it's gas initially. That term for that feeling is called quickening, and I don't remember it not showing up on the lab test. So know that word, quickening. About 6 months, the baby's going to start to get some eyebrows and some eyelashes. And things are going to kind of start to, all the big changes are going to start to change about here. So now the baby has done a lot of its development and everything is functioning, but we just need to get growing. So, and you know, there's certain things that don't work, right? Like the circulatory system. We need that lung bypass, and we're going to have a liver bypass. And that's okay, because who is the lungs for the baby? Who is making sure the baby is getting the oxygen it needs? Who's making sure that the baby's getting the nourishment that it needs? That's going to be the placenta. The placenta is the link between mommy and baby, and the connection to the placenta is the umbilical cord. Please remember from the vessels section that the umbilical artery is actually deoxygenated, umbilical vein are actually oxygenated. These are just showing different positions that the baby can be in. So the one on the left is not an ideal position. That's breech. So the baby's hiney is first instead of the baby's head. On the right, I have no idea what that's called, but I'm thinking that the baby's head is actually, the back is supposed to be up superior, and I'll find out the answer to that and I will let you know. But I'm thinking that's not a proper position. And then, what's going on here? We got 2 babies with 2 placentas. And actually, are they sharing one placenta? Maybe they are sharing one placenta. So if they're sharing one placenta, these would be identical twins. They have each their own placenta. Then they are fraternal twins. Identical twins, what happened with that is very early on when we're doing from 1, a zygote, so 1 to 2, 2 to 4, 4 to 8, 8 to 16, 16 to 32, somewhere in all that crazy division, there's a major separation. So it's super early on, the first couple of days during that initial mitosis, that the zygote splits into 2 separate little bundles. So before it even hits morula stage, so days, there's a major split. That would be identical twins. Fraternal twins, what happens with that is somehow by total luck, 2 eggs ovulated, so 2 ovum, and both of them get fertilized, but they're different. Remember, with all the genetic diversity from meiosis, the ovum are different, and of course the sperm are different, so they don't look alike at all. So fraternal twins is when you ovulated 2 ovum and you had 2 different sperm fertilize it. Identical twins is a split at that very initial meiosis. Okay, this is my excuse for being able to say that I taught y'all a little bit about genetics. Okay, what are alleles? Okay, you remember you get some information from mom, some information from dad. And during meiosis, we say that chromosomes line up with their homologous chromosomes. So there's going to be a chromosome that is about the same size from mom and dad, and they have basically the same information. So hair color, eye color, shape of your pinky, shape of your toenails, whatever. So they find each other and they line up together in pairs of 23 for meiosis, instead of all 46 of them lining up in the middle. Okay, when you have a gene from mom and a gene from dad, each of them is called an allele. Okay, so you got something from mom and something from dad. Then what we do is we set up this thing called a Punnett Square. Okay, and we divide what does mom have and what does dad have. So if it is a dominant allele, that would be the most common, most likely representation in general of the human population. So brown hair is dominant over blonde hair. Brown eyes is dominant over blue eyes. So what we'll do is if we say, somewhere in mom's family somebody had blue eyes, but mom has brown eyes, then her genotype means that she has both alleles somewhere in her body from mom and dad. And what we'll do is we'll set up this Punnett Square. If you have dominant and recessive, so the least common, it's going to be a capital letter and a small letter. Now, if nowhere in your family line do you have any recessive, whatever, there's no way you could have the genotype with a recessive, then it's both going to be capital letters. Okay, let's do blue eyes. If you have blue eyes, that's recessive. So to have recessive, you have to have both alleles recessive. So for example, I have bluish gray eyes. That's rare. So I would be 2 little A's. And then my husband's not a good example because he's also blue-eyed. But, if we put him in there, 2 little A's, what would all of our children be? Would all be blue eyes. Crazy, right? Okay, but let me do my parents. My parents are both brown-eyed, and there used to be the joke that I'm the mailman's daughter because I also had blonde hair. Now it's gray, so, whatever. I pick whatever color I want. But I was very recessive. My parents were brown hair, brown-eyed. But because they had a child with recessive, then we know that their genotype somewhere in there has recessive. So let's say if we had eye color of brown, but you have a blue-eyed child, then you'd have to have dominant allele, because the dominant's always going to win. If there's a capital letter in there, you're going to be that capital letter. But in order to have a child that had a recessive trait show up, then that means that somewhere in the parents they have the recessive trait. So my parents must have, let's do B's for brown eyes. So capital B, little b, 3 of their children by percentages, 75% of them would have brown eyes, because you take the letters and you drag them down to meet each other in the boxes. This box is called Punnett Square. But if their two recessive alleles get together during that meiosis part, they're going to have a blue-eyed child. My brother, brown hair, brown eyes. The term genotype versus phenotype. Genotype is what is on your genes. So my parents would be capital B, little b, both of them. I would have to be little b because I have blue eyes. Phenotype, that is actually what do you see. So my phenotype and my phenotype is blue eyes, my parents' phenotype is brown eyes. It's less specific. So although my parents are carrying the blue-eyed gene, the little b, you see brown. So their phenotype is brown hair. Okay, this is showing that two little W's would be a straight hairline. That's recessive. Most people have the tiniest little widow's peak. So that would be either both capital W's or a capital W with a little w. Let's look at some. Oh, look, there's an eye tray right there. That's what happened to me. Okay. Then on this one, right here on the right-hand side, this is going through traits that are dominant or recessive. So if you can roll your tongue, maybe you can roll it up like a tube, or maybe you can flip it over or whatever. It's a dominant trait. The inability to roll your tongue, that's a recessive. So that would be unusual. Earlobe attachment. Most people's earlobes are detached and free from their face. But some of them, not very many, it's a recessive gene, their earlobe will actually continue down to their face and they don't have a separate little dangly lobe. Arm folding. Everybody's going to do this now. Oops, I missed pinky. A crooked pinky is dominant. So most people are going to have a crooked pinky. Straight is recessive. Arm folding. When you fold your arms, does your right go on top of the left or does your left go on top of the right? If your left goes on top of the right, you're recessive. There's one in your lab book. It has to do with you interlock your fingers together. So you take your fingers and you interlock them together. Now, look at your thumbs. If your left thumb is on top, that's dominant. But your right thumb being on top, that's recessive. Anyway, you can read this and you can play with this and you get the idea. It's kind of cool. I mean, most people knew that blue eyes are not that common. Red hair is not that common. There's lots of different things that you can look up and see. Are you common or are you uncommon? Anyway, if you have any questions about that, please ask me during our little meeting during class time. But I'm going to sign out. That is your last lab. Genetics will not be on your lab test. I just threw it in there because it fits and it takes me 10 minutes. And then I can genuinely say for lecture, I have talked about genetics. And maybe the question would be something like, what is a Punnett Square? Or if somebody has two little b alleles, is that dominant or recessive? If somebody has a big B and little b, are they going to show a dominant trait or a recessive trait? As long as you have the capital letter, then you're going to show the dominant trait. Now your babies might end up having the recessive trait if your spouse has an allele with a recessive trait. All right, signing out. If you have any questions, I'll talk to you during our meetings. Y'all take care and have a very blessed Easter weekend.